Thank you for the introduction. Just wanted to touch base on as we move around and try to find different ways to stop the process of cartilage degeneration. And I want to focus today's talk on this emerging field, which has been very well defined in cancer biology, but not in osteoarthritis. And we're beginning to understand that this process of autophagy could be very critical in terms of maintaining the articular cartilage homeostasis and how cell death mechanisms as well as cell survival mechanisms go hand in hand to determine the fate of the articular cartilage. Now, of course, as we move along, we have to understand that joint homeostasis, of course, depends on not only these two processes, cell death and cell survival, but also other hemostatic functions. But before I get into talk about a little bit about autophagy, I'm just going to touch base a little bit about osteoarthritis. We know that it's the most common form of osteoarthritis, and I'm not going to go into details and how many people are affected. However, I just want to touch base that, of course, women are most affected by osteoarthritis than men. And probably this is the case of different homeostatic potential of women as opposed to men, which probably determine the fate of the articular cartilage. Now, of course, we all know the typical joint structures which are affected comprises of the articular cartilage. You have the subchondral bone and the synovium, which all are affected. And apart from these, you also have the fat pad and other surrounding joint structures which are also affected in osteoarthritis. Now, if you look at the pathophysiology of osteoarthritis, you have the articular cartilage, you have the surrounding synovial membrane, and you also have the subchondral bone, where articular cartilage, which comprises of only chondrocytes as only cell types present in the articular cartilage, can be stimulated by a wide variety of stimuli. It could be extrinsic or intrinsic stimuli, where once stimulated could further lead to a cascade of events such as upregulation of cytokines, wide variety of destructive mediators such as matrix metalloproteases, as we already talked about in previous talks, and ultimately leading to cartilage destruction. But not only chondrocytes, but other cell types such as macrophages, as well as synovial fibroblasts, which are in the synovium, also contributing to the disease pathology, where all leading to a cascade of event and then ultimately resulting in degradation of the articular cartilage. Now, when you look at the articular cartilage, you have the osteoarthritic cartilage, and then you have the normal human cartilage. Normal human cartilage, you have chondrocytes, which are nicely stacked. You have proteoglycans. However, you look at the osteoarthritic human cartilage, you have these cells which are undergoing cell death. This is apoptotic cells which are stained by tunnel staining. Also, you can perform a wide variety of other stainings such as caspase as well as part staining, which quantify the number of apoptotic cells in the articular cartilage, and that they all show the enhanced rate of cell death in an apoptotic cartilage. Now, the problem with articular cartilage is it's a postmitotic tissue. These chondrocytes within the articular cartilage know how to die, but they don't know how to survive. And as you're growing older, not your cells are actually also growing older. In other tissues, there is an ability of cells to actually restrain from the changing environment. They adapt very well. However, chondrocytes do not adapt very well to the changing environment. And like I said, they just know how to die. So how do we stop this process of cartilage degradation, which is the biggest million-dollar question we all have? And of course, that's the reason why majority of the regenerative therapies have failed, because chondrocytes ultimately do not respond to those regenerative therapies. Now, in terms of autophagy, this is a complex process where autophagy is required for maintaining not only development but also homeostatic process. In some cells, it can induce cell death. In some cells, it can induce cell survival. But most importantly, it is a cell adaptation process rather than cell death or cell survival mechanism as it's normally defined. Now, of course, autophagy can be regulated by a wide variety of stimuli, in which the primary stimuli could be hypoxia or nutrient deprivation, which is very critical in terms of articular cartilage. Now, of course, there are several biological roles of autophagy. Autophagy can be stimulated by a wide variety of stimuli, such as cytotoxicity, endoplasmic reticulum stress. However, mechanical stress is very critical in terms of osteoarthritis. And once autophagy is initiated, it results in suppression of autophagy. You have to have a very regulated autophagy process to be able to do the suppression of cell death and apoptosis. Also, it's involved in nutrient recycling. Now, the critical process, which I said, of autophagy is the cell adaptation process. And this adaptation process allows the cells to survive for a longer period of time, and as a result, cells can adapt. The process of autophagy is dysregulated during osteoarthritis, and I'm going to show how it actually is dysregulated. Now, within this autophagy pathway, there is a target called mammalian target of rapamycin. Now, it's a serine threonine protein kinase comprising of two complexes, TOR1 and TOR2. Now, if you delete TOR from C. elegans, you can double the lifespan of C. elegans. If you treat mice with rapamycin, which targets mTOR complex 1, you can increase or extend the lifespan of both male and female mice. So there is some kind of a link between mTOR, longevity, cell survival, and cell death. And that's what we wanted to actually look into. Now, how is mTOR and autophagy regulated during OA? Now, there are a couple of studies performed elegantly by Dr. Lotz from Scripps Research Institute in La Jolla. They actually showed that during osteoarthritis, the process of autophagy is compromised. That means when you have a degraded cartilage, its ability to survive is compromised, and this is probably due to not only the cells are dying, but there's something happening before apoptosis, which is that these cells are losing their ability to survive, and that's why they actually showed that there are critical markers of autophagy such as ULK1, which is the most upstream inducer of autophagy, is dysregulated, and as a result there is a compromised autophagy process in articular cartilage. They also showed that when you actually treat mice with rapamycin, you can induce autophagy process, you can increase an autophagic vacuole formation, and as a result you can reduce the severity of articular cartilage damage, which they showed in experimental osteoarthritic model. So taking from onwards from these studies, well, these studies came out, we were looking at how mTOR is regulated during osteoarthritis. So what we saw that if you look at the human OA cartilage, you have an increased expression of mTOR compared to the normal human cartilage. Similarly, if you look at OA human cartilage by immunohistochemistry, there is a higher expression of mTOR. And that actually is not present only in human OA cartilage, but if you look at dog experimental OA models, such as ACL injury model, as well as destabilization of medial meniscus model, mTOR is consistently upregulated in all these three species in osteoarthritic condition. So we wanted to go back and wanted to profile all the known autophagy genes which are probably regulated or dysregulated in osteoarthritis. So we compared osteoarthritic cartilage compared to the normal cartilage, and we performed these autophagy PCR arrays, and I don't expect you to look at this heat map. But what I want to point out is that out of all 84 genes which are involved in autophagy process, there were around 20 genes which were upregulated or downregulated in this pool of genes which we tested. But what was critical was that the genes which are involved in maintaining autophagy, such as ULK1, the most upstream autophagy inducer, as well as LC3, which is involved in autophagosome formation, are all dysregulated, or you can say downregulated, in articular cartilage in an osteoarthritic condition compared to the normal cartilage. So there's an overall compromised autophagy process in the articular cartilage. If you look at by immunohistochemistry, the number of cells which are expressing these autophagy factors is much more reduced in the articular cartilage, such as ULK1. It is highly expressed in the normal cartilage. However, it is reduced in the articular cartilage of osteoarthritic patients. And this, is actually not very clear from from the picture. But if you look at the PARP is a marker for apoptosis. You have an enhanced apoptosis, and you have an inverse correlation where ULK1 is downregulated. ADG5, which is another autophagy gene, is further downregulated, as well as BNIP3, which is an interactor with LC3B to perform an autophagic vacuole formation process, are all downregulated in an osteoarthritic condition. And this is, of course, consistent in dog models of osteoarthritis as well as mouse models of osteoarthritis. Now, of course, this is just, we performed in vitro studies, and we wanted to actually confirm the role of mTOR. And what is the exact role of mTOR in autophagy in vivo? And we took a knockout mouse approach, and we created a cartilage-specific mTOR knockout mice. First, we took an approach where we performed a germline inactivation, where we took Col2 Cree mice, where Cree is under the control of collagen-2 promoter, and specifically deleted mTOR in the cartilage. But this is in the germline. That means when these mice are born, they all have, they don't have any mTOR. What happens in these mice is that they are short in length, and they have defective endochondral ossification process. Their long bones are shorter compared to the control bones. So there is a developmental defect where mTOR is playing a key role in developmental regulation. So we wanted to go back, and we wanted to specifically elucidate the role of mTOR in osteoarthritis. So to be able to bypass the developmental defects, we took an alternative approach, and we created an inducible cartilage-specific knockout mice. That means that when mice which are born and they are around 4 weeks old, you actually give them doxycycline, and doxycycline is able to then delete or activate Cree and is able to delete mTOR transgene. And these mice were normal. They don't exhibit developmental defects. And then we subjected these mice to our mouse model of OA surgery, which is the destabilization of medial meniscus model. This model is a very subtle model as opposed to other models. It results into slow progression of osteoarthritis, which is more reflective of human OA pathogenesis. Now, if you look at the mTOR knockout mice, these are subjected to osteoarthritic surgery. What you actually see in these is the left-hand side is the control mice. You have the mTOR knockouts on the right-hand side. As you can see, 5 weeks post-surgery, you have loss of articular cartilage, you have loss of proteoglycans as expected, and loss of cellularity of the articular cartilage chondrocytes. And this phenotype becomes more profound at 10 weeks post-OA surgery. You have more cell death as well as more destruction of the articular cartilage. However, if you look at the mTOR knockout mice, are either at the 5 weeks or 10 weeks post-OA surgery, these mice are protected from osteoarthritis. Now, going back, mTOR is a master negative regulator of autophagy. That means when you delete mTOR, you're upregulating autophagy. So we wanted to actually check if that was indeed the case in these mice. So we took the sections from these control and knockouts, and this is an mTOR wild type. This is an mTOR knockout exhibiting no mTOR expression in the articular cartilage. When you perform tunnel staining, you have reduced cell death. That means the cartilage is protected in these mice. When you do PARP, P85 staining, which is another marker of apoptosis, you can see apoptosis is much more reduced in the knockouts, almost negligible in the knockouts compared to the controls. However, if you look at the ULK1, LC3B, two critical markers of autophagy are upregulated in these mTOR knockout mice. And then if you look at the MMP13 or C12C, which is a collagen breakdown product, their expression is much more reduced in the knockouts compared to the control. So there's an overall protection in these knockout mice compared to the control mice. Of course, we quantified by different parameters. Even if you look at the Western blot technique, you can see when you delete mTOR, you upregulate ULK1, you're upregulating the expression of or the conversion of LC3B1 to LC3B2, and so there is an upregulation of autophagy in these mTOR knockout mice. So we wanted to explore further what is the actual mechanism of mTOR pathway in osteoarthritis. And of course, the first thing which we wanted to test was rapamycin, which is a pharmacological inhibitor of mTOR, mTOR complex 1. So we took human OA chondrocytes and we put rapamycin in these chondrocytes, and we wanted to see if rapamycin is able to reverse the phenotype of human OA chondrocytes. Now, when you treat cells with rapamycin, you're able to block the mTOR signaling pathway, which is seen by this P70S6 kinase, which is downstream of mTOR. And by treating cells with rapamycin, you're increasing the autophagy process. As you see, LC3B1 is converted into LC3B2. Now, what is really important in this case is that when you treat the cells with rapamycin, you're increasing the expression of aggrecan and type 2 collagen, the two major extracellular matrix products. However, you're decreasing the expression of MMP13, the major catabolic OA product, as well as CCL5 and CCL2. So by giving rapamycin in these cells, you're able to decrease the catabolic process and increase the anabolic process. So we wanted to check what exactly is going on in these cells. Now, within the mTOR pathway, there is another protein which is very, very important, serine threonine protein kinase, which is ULK1. Whenever mTOR is upregulated, this ULK1 is downregulated. And ULK1 is actually responsible for increasing autophagy or initiating an autophagy process. So what we did was we took our cells and we treated that with rapamycin in the absence of ULK1 siRNA. And what we wanted to do was we wanted to see, of course, rapamycin is giving protective effects, but can these protective effects be reversed if you're blocking something downstream which is actually initiating autophagy pathway? Now, as you can see, you give, this is a control siRNA, you give the ULK1 siRNA. As you give ULK1 siRNA, you block LC3B conversion, which is important for autophagy induction. Not only that, when you actually taking rapamycin is increasing LC3B. However, in the presence of ULK1 siRNA, the protective effects of rapamycin are abolished. And as you can see, by giving rapamycin in the presence of ULK1 siRNA, MMP13 goes up, as well as CCL2 goes up, the major catabolic mediators, as well as if you see LC3B and ATG5, they are downregulated. So what we saw from this study was that probably in an osteoarthritic condition, and of course as you're aging and your circulatory cytokines such as IL-1 beta or matrix metalloproteases or any other kind of inflammatory or catabolic stimuli, they are directly resulting in upregulation of an mTOR signaling pathway, whereby these are resulting in blocking ULK1. If you block ULK1, you're blocking induction of autophagy. As a result, you lose chondroprotection. If you're losing chondroprotection, as a result, you are increasing the rate of apoptosis and decreasing cell survival in the articular cartilage. So we haven't stopped here, and we also have gone a little bit forward with understanding what actually could be directing mTOR in the articular cartilage. A few years ago, we were interested in this factor called PPR gamma. It's a transcription factor, and when you take PPR gamma knockout mice and you look at osteoarthritic process, the PPR gamma knockout mice exhibit accelerated OA compared to the wild-type mice, and this phenotype becomes more profound as as the surgery time increases. So you delete PPR gamma, you increase acceleration of OA, osteoarthritic process. What also happens in these PPR gamma knockouts is that as osteoarthritic phenotype is increasing, your rate of autophagy induction is also decreasing. So there is a correlation between cell death and cell survival mechanism, where knockout of PPR gamma is reducing cell survival and increasing rate of cell death. So we wanted to go back and look at if there was any relationship between PPR gamma or mTOR and try to delineate if there was any cell death cell survival mechanism which was associated. So we created a double knockout mice where PPR gamma and mTOR were both deleted in the articular cartilage, and then we subjected these mice to same model of osteoarthritis, which is the DMM model. Now, if you look at the control mice and look at the PPR gamma mTOR double knockout mice, now PPR gamma just single knockout mice exhibit accelerated osteoarthritic process. However, if you look at the PPR gamma knockout mice, again, by deleting mTOR, you're able to reverse the OA phenotype and you're able to rescue these mice from osteoarthritis. So indeed, mTOR is a critical target, which probably is, we don't know how well this target could be pharmacologically, but at least genetically, by deleting mTOR, you're able to protect the articular cartilage. Now, moving forward, what we're trying to do is we're trying to understand if ULK1 is indeed involved in pathophysiology of osteoarthritis, and if by giving ULK1, are we going to be able to protect our articular cartilage? And this is it's a fishing experiment. We really don't know what to expect from it, because articular cartilage is not the only joint tissue which is present in the joint. You have the synovial membrane, which is in a very different homeostatic potential. That means autophagy process could be downregulated in the articular cartilage. However, in the synovium, where you have these fibroblasts which proliferate so much, it probably have a very higher potential of autophagy process. So it's really essential for us to see, by putting in something which increases cell survival, what it's going to do in the joint. So it's a fishing experiments. Hopefully we have some results from here, where we're taking ULK1 adenovirus and putting them intra-articularly into the mouse joint, which are going to subject it to osteoarthritic surgery. What we're also trying to understand is if there is overall body homeostatic process is then resulting in joint alteration and joint homeostatic process as well. And what we're doing is we're trying to make mice obese or lean depending on the diet they are getting, and then trying to then understand if it is altering not only their physical functions and homeostatic process in the systemic circulation, but how by increasing the fat in the diet is altering the joint homeostasis, and how that alters the apoptotic process as well as autophagy. And this is being conducted by Alexa and Yui in my lab. And we don't want to stop at mouse studies or preclinical studies, but we're trying to understand how this actually stratifies into the human OA population, because autophagy could be a critical process, but unless and until we define in human OA population, we will never be able to justify what exactly happens. So what we're doing is we're trying to understand the regulation of autophagy process in all the joint tissues, from early knee-OA patients as well as from end-stage knee-OA patients. And we're trying to understand how, if we are going to have a therapy which is coming, which is targeting autophagy, could be beneficial, or it is just a fluke. So thank you very much. This is what I wanted to present. So thank you.
Questions?
Well, I didn't want to miss this, but that's why I came. That's why I came back. Do you think that inducing autophagy, you can control cell death?
To be honest, I don't know if we can control the cell death per se. We probably can increase the survival or adaptation of the cells. There is a very thin line between this process, which we always sort of probably goes hand in hand. But if we say autophagy is just priming cells to survive, which probably is not the case. There are some cells in which autophagy is initiated will undergo cell death. but there has to be an optimal level. But we don't know what is the optimal level. We really don't know autophagy is good or bad. We know that mTOR knockout mice have consistent phenotype. We have performed around 20 to 30 mTOR knockout mice or mTOR double knockout mice with PPR gamma. They all are protected. If you put rapamycin, they are all protected from the osteoarthritic manifestations. So.
Yeah. Anyway, this is very complex because in the joint, it's not only the cartilage. That's why we have to study also synovial fluid, synovial membrane.
That's exactly what we're doing right now. So what we're trying to do is we're trying to look at autophagy potential of different tissues. So right now, we have results from the synovial tissue as well. In the synovial membrane, if you just isolate fibroblasts, fibroblasts have a lot of autophagy in them. So they have all the markers which are downregulated in the cartilage are all upregulated in the fibroblasts. So that means there is a very different homeostatic potential between these two joint tissues. That's why TGF-beta, if you put TGF-beta could be really good for the cartilage, but TGF-beta may not be good for the synovial membrane because you are increasing the fibrosis of the synovial membrane. However, it could be really good because you're increasing the potential of the articular cartilage to manufacture extracellular matrix. So that's why anti-TGF or TGF-beta therapy could never work in a joint because of this.
In what model are you working with?
So in this case, it is destabilization of medial meniscus. So basically, you take the patella out and you transect the medial meniscus.
No, but in what model you said?
Medial meniscus model.
No,
mouse.
What animal?
Oh, so this is in mouse model, but we...
Mouse?
Yes. So we use mouse model, and we also use dog ACL model.
Because the mouse model is very difficult to have a synovial fluid.
Yeah, actually, this DMM model is probably, right now, is probably the best model to actually see the structural alteration. It's a very slow, progressive OA model, and has a very, very good... I would say around 60 to 70% reproducibility, which is very high in terms of osteoarthritis.
Because we're developing now a mini pig model.
Yes.
And there we can study the whole joint.
Yes.
And that will be very...
Absolutely.
Very good.
So, yeah.
Okay. Thank you very much.
Thank you.